Good morning, good afternoon, or good evening, wherever you may be in the world. Hi, my name is Meredith Burgess, and I'm one of the Regional Directors of Medical Affairs for Biomarkers and Diagnostics at Merck MSD. I'm here to talk to you today about our work around homologous recombination repair and genomic testing. This discussion is for your educational training and use only, and it's done in collaboration with AstraZeneca. Today we're going to talk about HRR and the tests related to detecting mutations for homologous recombination repair, and then ultimately we'll talk as well about the implications in clinical practice. Our core training objectives today are three basic buckets. First, describe and discuss the following: the basics of gene regulation, replication, and repair mechanisms, the role of HRR as a mechanism for DDR, or DNA damage response, including key genes that encode for HRR pathway proteins, multiple types of genomic alterations, from point mutations to large rearrangements or loss of heterozygosity. We'll also be able to compare and contrast tumor testing, ctDNA, as well as germline testing. In addition to genomic testing methodologies, we'll discuss such as PCR, finger sequencing, and genomic scar assays. And finally, we'll conclude with a deeper dive into the number of factors that impact clinical practice, critically analyzing those factors and how they impact the implementation of genomic testing into practice. We'll discuss a fit-for-purpose approach to selecting the right test, and also, again, those factors that impact the testing in practice. First, we'll anchor with the DNA damage response and homologous recombination repair, DDR and HRR respectively, and some of the core biology and biological principles in this science. First, I want to just touch base and remind us around DNA replication. The DNA replication is a very highly regulated multi-step process, but there are three key things to keep in mind or pull forward through our conversations today, and also in any other data, literature, other conversations you may have about DDR and HRR testing in the future. The three key things are for DNA replication. One of the core principles is that we have the two strands of DNA, and they're complementary in nature, meaning that when we have replication and we're utilizing the replication fork, as you see in the left-hand side of the slide, you see that there is also a complementary, or what would be a template strand, that we can work off of to replicate our DNA. That's a core piece of what we need to remember throughout the conversation today. The other piece is around all of the enzymes, which are really highlighted as the key proteins in the replication fork on the right-hand side of the slide. Understanding of how these enzymes work and process DNA and replicate the DNA with high fidelity, if possible, are some of the key scientific principles we utilize in PCR and other technologies that enable us to do the testing that we're going to talk about today. And the last key piece, other than having just the idea of the template DNA, is putting it all together. And that DNA replication, it has to be done in sequence. So each specific piece that's added, each nucleotide that's added is done in a very purposeful way, matching that template, and it's done in a very specific high fidelity way by those enzymes. Keeping these ideas in mind will really help us in understanding the deeper science as we go throughout the day discussion. The next key principle that we want to remind ourselves on is just about gene expression and the fact that it is a very highly regulated process as well. On the left-hand side of the screen, you'll see just a general depiction of the basics around gene structure. While the DNA certainly encodes for the specific protein, and there's a lot of message there that actually goes into transcription and ultimately translation, there's also coding regions, if you will, on either side and interlaced within the gene itself to determine what is an intron, what is an exon, what will be expressed into protein, what will not, and importantly, also the regulation of the transcription itself. So while it's a very complex piece and complex process, it's just depicted here in a simple simplified version, and it's really just to remind us that gene expression is highly regulated, and the DNA sequence, not just of the gene itself, but areas of DNA around the gene matter. On the right-hand side, the transcription to translation picture is just another good reminder to say that even if once you get beyond that level, differences at the single nucleotide level can actually impact not just transcription, but ultimately they could impact the translation of a gene into protein. Eventually, the folding of the protein function, and or its regulation of expression and utilization can all be impacted, and you can trace all of that back again to the single potential change in a nucleotide at the DNA level. So understanding DNA and replication, that high fidelity process, as well as the gene expression and how it's highly regulated and the multi-steps that are involved there, really come together to define the foundational principles we need to understand when we are assessing and understanding different mutations, how they fit into clinical practice, but also the tests we use and the technologies we use and how they come into play. We know that cancer develops as a result of different mutations in certain genes. Two of the key points that we have to keep in mind are that mutations can be germline in nature, as depicted on the top half of the screen. Germline mutations are inherited from a parent, and we have two copies of our genes. When we get that separate acquisition of a mutation due to DNA damage, we ultimately can have loss of function, which means the protein is no longer functioning regularly. And in the case of HRR, which we'll connect the dots to in a little bit, that's what we're really focused on later today. So the top half represents a germline mutation. The somatic mutations represented in the bottom half are those that arise from DNA damage caused by internal or external insults. And again, because we have the two copies of the DNA, acquisition of that second mutation is what truly leads to damage and ultimate loss of function. Repair of the DNA damage ultimately will help prevent carcinogenesis. And as you look at the slide moving from left to right, you see a list of internal and external insults that can cause DNA damage. The picture of the alpha helix really capturing the idea of that DNA damage response, which we'll talk about in a moment, and the fact that there's a multi-step process as an end result. Recognizing the damage, making the proper DNA repairs, ultimately regulation of cell cycle as well, and then the decision-making within the cell, whether it should go through senescence and ultimately potentially controlled apoptosis. Ultimately, it's the genomic instability manifested as the cell's ability to tolerate that DNA damage that's really a hallmark of the cancers. The goal, of course, is to prevent carcinogenesis by having active DNA repair mechanisms. We'll talk about those in a little more detail. DNA damage response, if you take a step back and think at a bigger picture, a bigger level, DNA damage response, referred to as DDR, is a very broad term. It really represents a holistic biologic process that goes from recognizing the fact that there's DNA damage that has occurred to regulating the cell cycle, bringing in the right proteins to get to the spot of the DNA damage, bind there, recruit additional proteins, go through any of the number of DNA damage repair mechanisms you see on the right-hand side of the screen, and then ultimately move into the next stage of the cell cycle into hopefully a normal cell restored DNA. The challenge, though, is because we have multiple ways of repairing DNA damage, if there's multiple hits, if there's multiple pathways that are impaired, it can ultimately lead to cell death. So you can see on this slide, as you're moving from left to right across the top, you see the representation from a normal cell. If you have a cell that's homologous recombination repair deficient, if you have a cell that has single-strand base excision repair deficiency, or a cell that represents the combined deficiency of both single-strand repair as well as double-stranded break repair. And as you see kind of depicted almost from this table, checks and X marked representation, you can see that if you have just homologous recombination repair deficiency or the single-strand base excision repair deficiency, the cell can still potentially survive. But ultimately, when you have an impairment of double-stranded break and single-stranded break repair pathways, that can ultimately lead to cell death. Again, a moment ago, we spoke about there are different pathways for DNA damage to trigger breaks in the DNA, and then ultimately different repair pathways. This is just giving you a simplified version. A reminder that single-stranded breaks on the left are ultimately repaired by base excision repair, and PARP plays a role in this pathway. Bulky adducts can be repaired by nucleotide excision repair. Base mismatches, insertions, deletions, those are typically repaired by mismatch repair, MMR. And then double-stranded breaks, the most lethal type of DNA break that's the hardest to actually overcome and repair, can be done by homologous recombination repair, which is our focus today. It's a higher integrity, higher fidelity repair compared to non-homologous end joining, where more random bases are added. Again, recall that PARP enzymes participate in the single-stranded break repair pathway. So when you have DNA damage that causes the single-stranded break, PARP plays a role and comes in to help repair during the single-stranded break function and ultimately restore cell function and cell survival. Homologous recombination repair, on the other hand, as we've already discussed and highlighted, is really focused on double-stranded breaks. We're not going to get into all the details of the actual repair pathway here today, but you can see the picture on the left-hand side showing you the replication fork area, looking at the template strand, identifying, the double-stranded break, and going through a series of molecules and recruitment of proteins to take out that double-stranded break and repair it. But one of the most core things to know is that the right-hand side of the slide actually walks you through a number of key terminologies like loss of function, HRD, LOH, etc. So when you have a moment, it's good to take a moment to actually read through those. We're going to go ahead and move forward, though, as we utilize some of these terms in the next couple of slides. When you look at HRR as a whole, we know that mutations in the HRR pathway, especially BRCA, which is the most commonly discussed type of mutation, and BRCA1 or BRCA2 genes, they're found in a wide variety of tumors. And you can see this depicted here by the stacked bar graph. Moving from left to right, the mean total level of HRR deficiency mutations in a whole range of different cancers. For our purposes, we're going to focus and connect a little bit more today around ovarian, breast, prostate, as well as pancreatic cancer. And you can pause and take a look if you'd like. The genes represented here on the right, they are all represented within the HRR pathway. And just to call out again, BRCA1 in the red and then BRCA2 in the slightly darker blue color are the most common of the HRR genes that are mutated and known to play a role in carcinogens. So again, BRCA1 and 2, we know they're the most common mechanism for homologous recombination repair deficiency. And you can see this depicted here, just kind of the role that they generally play within the repair pathway. So when you have the double-stranded break on the left-hand side and you move across the side, we see the recruitment of APR and ATM. Ultimately, you get the connection to BRCA1, which will help recruit and signal for other proteins to come into play. Downstream of that is BRCA2. And ultimately, you do get the repair of the double-stranded break. This slide, the 15 HRR genes and their function slide, as well as the next slide, are a two-part table where we list 15 of the common HRR genes where we have known mutations that impact different types of cancer. And these are the 15 genes of interest to us where we have done testing, particularly tumor-based testing, to assess HRR gene mutations. I'm not going to get into detail on each of these genes, but they're listed in general prevalence. And as well, there's a description of their brief protein function and some of the example types of mutations that you can find associated with these genes, such as point mutations, large rearrangements, or silent mutations. This is the second slide of that table. And now I'm going to take a moment to sort of summarize where we are so far. The failure of cells to repair the DNA single-stranded break or the double-stranded break can lead to that genomic instability and ultimately carcinogenesis. Again, moving left to right, this slide is really important because it really connects the dots, if you will, from the actual insults that cause DNA damage, whether it's external, such as radiation, or an internal, such as a spontaneous mismatch with a mutation or a mispaired base. Ultimately, what you have can be either single-stranded breaks or double-stranded breaks. And then the repair mechanisms that are triggered as a result under the DDR. PARP can play a role directly with single-stranded breaks, and then you have noted on the slide for you the indirect role, if you will, that PARP can play for double-stranded break repair. But importantly, when you home in on homologous recombination or homologous recombination repair, noted as HR or HRR, you see the genes that we've been talking about and the genes that we'll continue to talk about today, particularly BRCA1, BRCA2, BRCA51, ATM, etc. PARP plays a role as it contributes to the fine-tuning of homologous recombination, as noted. Ultimately, though, through the process of DDR recognizing the DNA damage caused by those insults, the type of breaks that you have, single-stranded or double-stranded, the end result remains relatively the same kind of process. You go through a minor or a major DNA repair, the cell cycle has been arrested. You can have a decision to permanently arrest the cell cycle if there's a lot of damage. There's transcriptional regulation of the proteins. There can also be senescence and ultimately potentially apoptosis. And then hopefully that what you ultimately have is complete repair of any DNA damage and ultimately restoring the cell so that you have prevention of carcinogenesis. So now that we've anchored on the idea of DDR, DNA damage response, and talking about the HRR, homologous recombination repair, mutations of interest and proteins, we're going to dive deeper now into actual testing and looking at the types of mutations for HRR genes that are of interest to us. First, just a couple of key examples. I would encourage you also to utilize this resource noted here as a reference for the slide, as well as any other resources throughout the presentation to dive deeper, also to look into the details for some of the figures. But you'll note here that there are a number of different types of genomic alterations, and some of these really stem from just the change in some manner of a single DNA base. They can range from a missense mutation to a nonsense mutation, insertions, deletions, often referred to as indels, a repeat expansion, or frame shift. The definition for each of these is included here, but the main take-home is that even a single nucleotide-based change can ultimately have an impact. Thinking bigger picture than that, though, if you take a step back, you can also find duplication of larger sections of genomic material, larger sections of DNA that gets replicated, a larger duplication, or you can find changes more so at the chromosome level. And in particular, some of the bigger things would be copies of huge sections of chromosomes, or literally a loss or an excision and complete loss and deletion of large chunks of one chromosome. And ultimately, too, leading to loss of heterozygosity, which is somewhat pictured on the lower right-hand corner. But ultimately, even if you have a single DNA-based change, or you have some of these larger alterations, At the end of the day, we want to understand how these changes actually impact the function of our HRR genes and therefore the function of being able to repair double-stranded breaks. And we also need to understand them so that we know how to best test and identify these types of alterations. As we've already noted before, the HRR pathway is certainly very complex and involves a series of a lot of different proteins, many of which are pictured here. And again, the idea is moving top to bottom, looking at that double-stranded break, identifying it, going through the systematic process of trying to have a high-fidelity repair as much as possible to not generate any additional mutations, but to truly repair that double-stranded break to a wild type or a normal DNA. Deficient homologous recombination repair can actually be detected using multiple different methods, and it depends a little bit on what type of question you actually have as to what type of approach you take. So we're going to take some time to walk through this slide a little bit together. So let's go in sequence, going from the top to the middle and then ultimately to the bottom. So across the top, you'll see the genomic perturbations as well as the assays. If you think about it at the very minute genomic level, looking at the actual DNA strands themselves and the individual bases there, you ultimately could have a change there that leads to altered gene expression. You could have the somatic gene mutations. You could have germline mutations. So looking at the DNA and some of the transcription level of what's actually happening, you can assess this by doing sequencing. You can sequence DNA. You can also sequence RNA. But the main approach here is really focused on a sequencing approach where you're systematically taking that genomic piece of information, that genomic code, and you're systematically calling out each nucleotide base. In the middle, across from deficient HRR, deficient homologous recombination repair, you also have the assay approach of real-time HRR assays as well as gene expression profiles. And these are almost a segue or a connection, if you will, from that actual genomic information meant to be captured by sequencing up above and moving into the more functional component, if you will, or outcome component captured by signature approach or genomic scars below. The deficient HRR bucket of changes, if you will, and the assays related to that can take on multiple different components and come at it from multiple angles, but the main point there is you're really trying to focus on the transcription and the regulation piece that gets impacted from the genomic change and actually looking at how those proteins in HRR are being expressed and regulated. That's the main focus for the deficient HRR and those gene expression profiles and real-time assays. The bottom section is a little bit more cleaner and straightforward to understand in that it's really looking at the results of an overall HRR deficiency. When a cell cannot repair all of those double-stranded breaks, their natural outcome is then to have a lot of double-stranded breaks accumulate. And when you don't have HRR as a pathway, you have to use non-homologous end joining as a solution. And when you do that, you introduce a lot of random non-homologous end joining base units, which means ultimately you're creating a lot of additional mutations and a lot of scarring, if you will, for the DNA. And that you can detect basically using a signature approach to identify those types of scars across the DNA. So overall, this figure just kind of represents and walks us through the fact that deficiency in HRR you can capture at different levels within the cell function, and based on how you're capturing that, you utilize a different methodology or approach to look, whether it's the genomic level, actual coding level, you use sequencing. If it's more in the transcription level or translation of the proteins, you look at the real-time or gene expression profiles, and ultimately you can look at more holistically for a signature or a pattern that results from having HRR deficiency, and that shows up as genomic scars. For this next slide, it's the pathogenic BRCA1 and 2 variants, and it's a depiction of the general BRCA1 and BRCA2 gene structures. And what the main point here is just to show you and draw your attention to the fact, again, remember early on we talked about all of the regulation for DNA replication, but also gene expression. And one of the key points about BRCA1 and BRCA2 proteins is that each protein, of course, has a lot of different specific functions and domains. And what you see in the genomic level here is a lot of specific domains that have been known and clearly identified for both BRCA1 and BRCA2 as an example. When we look at these particular examples, moving to the next slide here, when you look at mutations for BRCA1 and BRCA2, you find that there are different types of mutations, both either germline or somatic. They can be nonsense, splice variants, frame shifts, etc. But you notice they are across and along the entirety of these genes, for example, but they also seem to potentially cluster in certain areas. There's more research going on to better understand this, but the main takeaway is that when you think back to the actual structure of the gene and that each of those sections of the gene matter in a different way to the folding of the protein or the function of the protein, the binding of the protein, etc., now translate and overlay all of these mutations and alterations, you realize that each of these individually can have significant impact to the function of BRCA1 or BRCA2. Indeed, a majority of the BRCA1 and BRCA2 mutations are actually point mutations, meaning they're usually single nucleotide changes. Again, anchoring back to one of the first things that we said, it's really important that the template DNA component is critical, but also that the sequence really matters. Each individual nucleotide matters. The insertion or deletion of a couple of small numbers of bases together with the single nucleotide changes, represents over 90% or so of the impacting protein function changes. The remaining almost 10% for BRCA1 and BRCA2 tend to be larger rearrangements of DNA segments, and it's challenging at times to detect those changes, but they are depicted here in the schematics below and to the right. Knowing that the DNA changes that we have matter not only to the protein, but also they matter to the type of testing that we use or the methodology and approach that we use. It requires different types of sequencing methods. If you have a single nucleotide change, then one of the testing methods could be finger sequencing, or more current testing that's more predominantly used would be next-generation sequencing approach. If you have a small insertion or deletion event, commonly known as an indel, then you can also use a similar type of approach. But again, calling out those large genomic rearrangements, if you have a larger piece of DNA that's been changed, been missing, excised, duplicated, et cetera, those bigger changes are harder to detect using finger sequencing and sometimes NGS as well. But it's really the PCR-based techniques where you can use a very specifically targeted approach that really is employed more commonly to detect those large genomic rearrangements. Some of the common ways to detect BRCA mutations would be the sequencing technologies. We've touched on a little bit of finger sequencing. You've probably heard of it mentioned, and typically when people picture sequencing in their mind, they're often thinking about finger sequencing as it's really the predominant original type of sequencing approach. And then we also have next-generation sequencing, which was really developed to try to improve, the turnaround time, but more so just the idea of bringing forward a high-throughput resolution assay that could actually generate a large volume of data and run multiple clinical samples. There are trade-offs and changes whether you do finger sequencing or next-generation sequencing, and we'll talk a little bit more about that in the next couple of slides. Here in this slide, you see the direct advantages and disadvantages in comparison between finger sequencing and NGS. Finger sequencing is typically referred to as being the gold standard. It's a very, if you will, almost systematic way of going through and cataloging and identifying each specific nucleotide. And it's relatively widely available in relatively simple workflow, but the challenge is that it has a very limited throughput. You can't run as many samples at the same time. That's making it also less cost-effective, and it can be challenging to detect the large gene rearrangements, which are usually more so detected by PCR methods. Next generation sequencing, much more commonly used. Most commercial tests used around the world and also research-based tests used around the world are predominantly an NGS mechanism or throughput. It's to use high throughputs. You can run a lot more samples. You can multiplex. It usually costs less than Sanger sequencing. It can be much more automated, et cetera. And in addition to that, it's good to note, particularly, it's less sensitive for those large changes over 20 base pairs. And it can also potentially miss some mutations that Sanger sequencing would have identified. In summary for this section, just a real quick note on the NGS, workflow, if you will. It goes through quality assessment, so it can remove, trim, or correct and readings that do not meet the defined standards. You can do a read alignment, variant identification, and we're going to talk in more detail, particularly about variants and how they're defined, annotations to filter and prioritize the potential disease-causing mutations for further analysis, and then ultimately you have visualization where the display aligned reads, you get the mapping with the quality and identify mutations combined with the annotations from the public sources. We're going to highlight some of those key points next in the clinical section. For this next section, we're going to get into more of the clinical considerations for HRR testing. And please keep in mind as we work through this section that some of the key points may or may not apply as much to your specific country. So keep in mind your local needs and how clinical practice is carried out in your region. We know many human cancers have been associated with homologous recombination deficiency. There's a whole bunch of them listed here. And in particular, when we dive down and look specifically at BRCA mutations, we know of particular interest to us are certainly breast cancer, ovarian cancer, pancreatic cancer, as well as prostate cancer. Some of the prevalence numbers are reflected here. But keeping in mind, for many of these, we have very limited data on overall prevalence, and the prevalence may change depending on the populations worldwide. Narrowing down a little bit more specifically, when you look at breast, ovarian, prostate, and pancreatic cancers, there's an overall lifetime risk for developing cancer in the general population, and that's depicted in the left-hand column under the gray area. For patients who harbor a BRCA1 or BRCA2 mutation in the middle and right-hand columns respectively, you can see that the cumulative risk by age 60 to 80 actually increases if you have one of these types of mutations relative to the overall general population. And because of this, it's more typical that HRR gene testing, and in particular for BRCA1 and BRCA2, the testing is often done through guidelines or just in general practice is completed based on trying to assess familial risk or germline mutations. One of the key points, though, that we touched on earlier as we talked through the different types of approaches to identifying HRR mutations is the fact that there are causes of HRD and then there are the effects of HRD. And we can utilize testing approaches to look at either of those. On the left-hand side, if you look at BRCA mutations specifically, thinking in particular germline or tumor testing, or more broadly looking at all of the other HRR genes, you're looking more at a gene panel where there's multiple genes being examined in addition to BRCA1 and BRCA2. One of the main key points there is that really that's targeting the causes of HRD. So if you're doing a test, a genomic test, looking for germline or tumor, specifically for BRCA1 or 2, and also any of the HRR genes, including those 15 genes specifically listed in our panel developed by AstraZeneca, those are all looking for in testing for causes of homologous recombination deficiency. The effect of having an impaired homologous recombination repair mechanism ultimately leads to that genomic scar, which we touched on earlier, and also can lead to patients having a more durable and repeated sensitivity to platinum-based therapies. Looking at the genomic scarring test or that clinical enrichment with the platinum sensitivity, both of those are, if you will, tests, or in the platinum case, a surrogate marker, if you will, for the effects of HRD. So taken together, you can see that when you look for homologous recombination repair deficiencies, you have multiple areas to think about and different ways to think about going about testing for patients and identifying patients who have the homologous recombination repair deficiency in practice. Currently, we are really undergoing what could be considered a paradigm shift in how clinical practice will manage that homologous recombination repair deficiency testing and the approach on how to identify those patients who have that deficiency. Moving left to right across the slide, most of the references and key points on this slide are related to ovarian cancer as well as breast cancer, where we have a more rich history and more abundance of data supporting our understanding of, in particular, germline mutations. But what you really find is the assessment before for mutations was really looking at the predisposition or the prognostic value, identifying the familial risk and the prognostic impact of these mutations for specific patients as well as for their families in the case of familial risk. Moving forward to where we are now and going forward into the future with PARP inhibitors, the interest around understanding the HRR deficiencies and the specific mutations more collectively is to understand a truly predictive clinical decision. If we get these results, will we be able to then, based on these results, predict the patients that could respond to particular types of therapy? And that is the predominant paradigm shift that we're seeing right now unfold in clinical practice based on clinical trials and evolving data and scientific knowledge. This is a great segue to this moment, which is where we should recognize there are a multitude of reasons for testing for HRR in the clinic. The first core reason is indeed understanding that familial risk. We've already touched on that. And again, if your main driver is understanding familial risk, you're going to want to do a germline-based test focused on determining germline mutations. There's also the interest in estimating disease prognosis, which is not necessarily one of the main drivers. Depending on your country, particularly if there's a lot of access or more common access to clinical trials, identifying patients eligible for trials may be a main driver for HRR testing. There may also be an evaluation of genomic alterations for other research purposes, such as creating a registry, identifying prevalence of genes, identifying responses to therapy. There could be a lot of additional research work that goes into and utilizes the results of HRR testing, but not necessarily informing patient eligibility for a trial. And lastly, which is where we're seeing the paradigm shift, would be HRR testing to inform and or guide treatment decisions. A multitude of organizations and guidelines impact HRR testing and inform clinicians and other stakeholders of what the clinical, if you will, gold standard or what good best practices look like. Within the U.S., certainly NCCN as well as ASCO plays a key role, in addition to AMP, Association for Molecular Pathology, and ACMG, the American College of Medical Genetics and Genomics. Outside the U.S., you may also find guidelines in other organizations such as ESMO, and also very specific organizations tied to either your country or also your particular tumor type that could play a role in posting guidelines or other commentaries such as white papers, working group publications, et cetera, that really could guide what key decision makers believe is the standard for testing in your country. If you need additional information, then I would just encourage you to go to your local resources and to better understand that and get the idea around what is the reason for HRR testing, what's the rationale that they're saying to use the test in the first place. Understand if the guideline or organization is recommending you do germline and or somatic mutation testing, And importantly, when is the testing being recommended? Is it something that should be done early on at the stage of diagnosis, or is it something that should be done later on during the course of care? And lastly, which genes are actually included as a benchmark for what you should be testing? Is it specifically calling out BRCA1 and BRCA2, or is it more of a broader recommendation or guideline that includes broader genes beyond just BRCA1 and BRCA2 to include more genes in the HRR pathway? Currently, when you look worldwide, we know that access to testing and you know, looking at different testing methods certainly vary region to region. But as a general approach, if you're looking for germline mutations versus somatic mutations, you can see there that blood is a good source predominantly for germline testing. Tissue sources can yield information on germline and somatic mutations. ctDNA, while it's taken from the blood as well, that approach to testing can give us information on either germline and or somatic mutations, and then ultimately CTCs, or circulating tumor cell tests. Keep in mind that the predominant type of testing globally, worldwide, across regions, if your region does have testing, it's most likely going to be a blood-based test for germline mutations. And then moving left to right, you can see that ultimately the CTCs are certainly not commonly performed and are much more of a research purpose or use. And tissue is really sort of the second one to follow behind germline testing based on blood tests. But again, you'll want to focus on and understand what's going on within your region as to the types of tests that are actually utilized in the clinical practices where you are. For the purpose here, we're using the FDA as a local example for what is important within the U.S. region, but this can also apply to your region as well, depending on the regulatory environment with which you work in. The main point here being that there's sort of a juxtaposition in any region potentially of the types of tests that can be used. And again, thinking about your region, it's important to understand what might be most directly relevant to you, but the principles remain the same. And that is that we can have, if you will, a true companion diagnostic or a regulatory path that's similar, where the diagnostic is approved in some manner and tied to our test and to our label and our data. On the other hand, there can be, in the U.S., it's a CLIA-validated LDT. In Europe, for example, we have a lot of LDTs as well. But there can be a situation where you have a broader access to types of testing that could be regulated differently. But just the call out for this slide is if you're in the U.S., a lot of this information does apply to you. If you're outside the U.S., this is just a call to stop and pause, understand your regulatory environment, and recognize that there may be situations where some tests are more highly regulated and approved, if you will, versus others. And it's something to keep in mind as you have conversations and navigate the landscape. Ultimately, though, the goal that we have is to really understand the science, understand our mutations, understand the role they play in the clinic, and understand our options to test, therefore identify those mutations that will be most meaningful to inform treatment decisions. And really, I kind of like to think of it as a fit-for-purpose approach, because tests are developed with a very specific purpose in mind, to identify very specific mutations or genomic alterations. And if we choose the right diagnostic, then ultimately we'll be able to better utilize those results to inform patient care. Real quickly to highlight a couple points on this slide, it's a flow chart type of diagram. And as you move left to right, you see that what's supposed to be captured is really the steps of going from a clinical question to understanding your test selection, getting the results after you've implemented that test, and then ultimately using those results to inform a clinical decision. The top part of the flow chart, though, is reflective of a predisposition or a prognostic type of test. You're really driven by a clinical question to determine patient risk for cancer or the prognosis. You're most often going to probably choose a germline test, and that's because you're really working to assess patient risk and ultimately the risk for the family. When you get the implementation and thinking through the results, you're going to be using a blood sample or buccal mucosal swab. The turnaround time could take several weeks, and in the case of the U.S. and other places potentially around the world, genetic counseling can play a significant role. And then ultimately, when you get the information, you're looking for prognostic indicators. It can indirectly impact the treatment decision, but also you're looking for potential risks for family members as well and additional follow-up. If your clinical question is like denoted at the bottom, which is much more predictive in nature, you're really trying to ask a clinical question to determine patient status and predict the response to therapy, so you want to be able to inform your treatment decisions. When you go through the test selection, you have to weigh, do you want to look at a germline test and or somatic test, where you can identify both types of mutations. And then ultimately you have the choice of taking either a tumor sample, which can collect germline or detect, germline, as well as somatic mutations for BRCA, for example. It has a shorter turnaround time in general from end to end, but it is important to reflex to a germline test if you get a positive result. On the other hand, if a decision is made to go with a germline test, you're only going to need blood or buccal mucosal swab. The turnaround time, though, can take several weeks, and genetic counseling is required and adds to that turnaround time. Ultimately, though, at the end, the clinical decision, in this case, you are asking the question to inform treatment decision, and hopefully the results that you get would determine eligibility for therapy. It directly impacts treatment in this case, and there will be a need for family testing if you did get a germline positive result. So you can see here that it really anchors on the clinical question you're trying to ask. And as you go through the steps to assess what test selection you should make, and then ultimately implementing that test and the results, in the end you land with a clinical decision-making point, and hopefully the test that you've chosen will directly inform the clinical decision you're making. There are a lot of considerations for picking that right test for the fit-for-purpose approach. We touched on germline testing as well as somatic or tumor testing that can capture both somatic and germline mutations. But the availability of sample is really a crux that will impact clinicians as they're working across different tumor types. Some tumor types do not have tissue samples as readily available as others, so they may by default be going to the germline-based test because of the availability of a blood sample or a buccal mucosal swab. There are also, of course, technical limitations to the specific test. We'll touch base on that in a little bit. In addition to just overall test availability and access, there can be a number of components that impact that piece for the clinical implementation of testing. And turnaround time does have a significant impact. If you're sitting in the clinic and you need the results as soon as possible, to inform treatment decision, then it's very likely you want to try to get the quickest turnaround time as possible. And then ultimately, one of the other considerations is what is the ability to interpret and utilize the results from a particular test. We know that not all tests report the results in the same manner or fashion, and sometimes it can be confusing. Despite the efforts to try to harmonize and have great standards around reporting of results, there's still some challenges to understanding the reports. So we touched on this before, but the availability of samples is a real key component for clinical considerations and how you're going to implement a test. In the case of BRCA testing specifically, it can be performed with blood, buccal mucosa, or tumor samples. Ultimately, though, wherever your sample comes from, the key piece is that it gets selected, we extract the DNA from the sample, and we do the DNA analysis. Keep in mind, though, that tumor samples are the only way to identify both germline and somatic mutations. When you have tumor tissue, it does matter how you handle the tissue, and we know this well. Whenever you freeze tumor tissue to adhere to the slide, you cause a lot of other challenges related to the integrity of the sample, and ultimately it can cause a lot of problems with getting a high-quality assay and results. The formaldehyde-fixed and paraffin-embedded method is much more conducive to getting the better results with tumor-based testing. We know that when you do a tumor-based test, that often you cannot definitively distinguish between germline mutations or somatic mutations, but rather the tumor testing result will tell you that you have a known mutation, suspected mutation, but in order to really be able to confirm that it's germline, you would have to reflex to do a follow-up test. There are some ways that you may hear about referenced in your conversations or in the literature or by your laboratory partners that talk about informatic subtraction, and there are some technical ways to get at understanding the resulting fraction that's somatic mutations, but overall, the take-home message is that at this point in time, tumor testing does not specifically call out and differentiate between germline and somatic. This next slide just looks at and compares the germline testing and the tumor testing, and it breaks it down for you in this table by sample type, the DNA analysis that can occur, the clinical relevance of where we are at this point, and also, of course, the familial implications. The main take-home points, again, that we've been touching on throughout this process is that deciding whether or not to do a tumor test or a germline test, you really want to be able to be informed on all of these points so that ultimately you pick the test that can help address your clinical question. In this next table, it's comparing when you look at germline or somatic homologous recombination repair mutations versus genomic scars or the chromosome structure, which is sort of juxtaposing, if you will, the actual mutations themselves versus the output or the results of the mutations in the scars. You can see the different approaches there, simply calling that out. For the most part, though, the work that we do focuses predominantly on germline or somatic mutations, meaning that we're really truly focused more so on next-generation sequencing and targeted gene panels. You see some examples there, as well as some pros and cons across the different types of testing approaches. And again, this slide as well offers as a resource for you to look at different HRR deficiencies, the types of approach, again, circling back to some of the slides we touched on earlier, comparing the methods, the types of examples that exist, and then again, the pros and the cons. So this collection of a multitude of different tables are really a main resource for you as you have questions to navigate tests in your landscape when you're looking at clinical trials data to understand the pros and cons of what approach they've taken and other things in the literature, just to kind of put things into context. For just a couple of slides, I want to touch base on ctDNA and recognize that it is still really sort of an up-and-coming type of technology. It's much more so really used for research purposes, and certainly we know that there are a lot of advocates for advancing the use of the cell-free DNA, the ctDNA approach. But for our purposes today, we're not focused specifically on ctDNA in this case at this point in time. But I do want to provide some information and background to give it context. So folks who are looking for the cell-free DNA approach or ctDNA approach recognize that the cell-free DNA, that approach is utilized in other applications within medicine. But really, the cell-free DNA can come through apoptosis, necrosis, or secretion into the bloodstream. And what you can utilize on the far right, you see in the bloodstream the representation there. You can get DNA from the apoptotic bodies. You can detect point mutations. You can look for copy number alterations, rearrangements, methylation changes, and exosomal DNA. But ultimately, the core takeaway is that there is a lot of cell-free DNA in our blood, and the relative representation of tumor cell-free DNA within that can change and fluctuate over time. The table represented here is a comparison of technologies for the analysis of ctDNA. And the main takeaway here is really just that you can take different approaches. You can say kind of we want to look at a general picture. We want to get a high-level idea. That's the candidate gene analysis. That's a much more rapid approach. The challenge, though, is that deep sequencing is really needed to really dive in deeper and understand specific mutations. And the challenge, though, of course, is that that would take longer time. Ultimately, though, when you think about ctDNA, there's a lot of standardization that's needed to bring it to the forefront so it can be utilized across multiple clinical situations and utilized by multiple labs. There's a lot of challenges to implementing ctDNA on a greater scale. And for many of you in countries across the world, you'll find that as you think about all of our work collectively together in HRR, that you may not be advancing or having conversations really about ctDNA within your country for clinical use, whereas some countries, it may become a more specific topic in the near future. The key point, though, of this slide is just to orient you to the fact that there are a lot of components, if you will, for pre-analytical variability as well as analytical variability that impacts the ctDNA analysis. They're both listed here, but one of the key points is really just thinking through the complexity of ctDNA, and not that other testing approaches don't have that challenge, but it is sort of a different challenge in that way. And indeed, because of some of the technical challenges, including just the availability of specifically ctDNA and the cell-free DNA taken from the blood, the ability to implement ctDNA reliably and reproducibly and accurately in the clinical setting has not really come to fruition. So now that we've talked a little bit about different testing methodologies into practice and talked about the trade-offs about different testing approaches and also just highlighted a couple key points about ctDNA, really want to move into a conversational discussion, if you will, here about some broader topics that impact implementation into practice. And one of the key pieces is actually the reporting of the results. So it can be really challenging to standardize the actual types of results that are given, how the tests are evaluated, the specific mutation, what is called something that's clinically meaningful versus not. And then in addition to that, you add the complexity of literally how a report is written and the format and the language that is used. And that can vary not just, country to country around the world, but it can vary from test to test and sometimes even from laboratory to laboratory. So while there have been great strides to try to harmonize or create standards to make sure that it's more easily translatable across tests in particular, it's still a challenge. And what you'll find is that even though there, again, is some things here about best practices, as noted here about things the report should state, like likelihood of false negatives, especially if the tumor content is insufficient or low, there's still a lot of things that are not necessarily called out in a report, and it can be really misleading. So it's important that we understand those limitations to each type of test and how the information is reported and for the results. But ultimately, when we're also communicating externally, we have to make sure that not only we understand it, but our stakeholders also understand these limitations as well. Here you see an example of the BRCA analysis companion diagnostic sample test page showing the cover page of the results. You can see just visually looking at it, it's relatively straightforward and easy to identify different parts based on format within the page itself. You can see clearly called out near the top that it's positive for a deleterious mutation, so it's already calling out there's a potential call to action for the clinic perspective. You can see this is kind of, if you will, an example of what might be considered a good standard, but again, calling out the fact that this is not what every test and what every company utilizes. So you can imagine that when you're getting multiple test coming in from different laboratories, different testings, it can be quite cumbersome to juggle all the different formats and such involved. But at least this picture gives you some idea of the type of information that is included in a report typically. And again, sometimes even though we've had great strides to make almost a gold standard or an expectation of what should be included, it doesn't guarantee that every test result and report will include all of this type of information. As a result, not only are the reports themselves challenging to navigate sometimes, but the results that are actually on the page, if you can focus just on those pieces, they can still be challenging. It's hard to understand if you have a true positive that, yes, a deleterious mutation could be identified, and if it's a true negative, it should say that there's no deleterious mutation identified. But what if it's indeterminate or it's uninformative? It could state that no mutation has been detected after the full sequencing. The carrier status of the family member is probably also negative or unknown, but one of the more challenging ones would be the inconclusive result, where there's a variant of unknown significance. And while all of these components, call information and can be informative to the clinician, part of the challenge, though, here is that you notice here there isn't a lot of information on the technical failures. So there could also be, of course, a technical failure for the test, and understanding how that might be reported on the report is also very important, because some tests will say that there's a negative result, and that negative result may actually be due to a technical failure where there wasn't, for example, enough DNA. So it may not necessarily reflect that it's a negative result for running the sequence and not finding a mutation. It could be that a mutation could not be found due to a lack of appropriate sample volume. So it's really important to kind of keep these things in mind, and one of the key pieces is that because of the complexity in certain countries around the world, genetic counselors are involved and get into those details to really understand the type of test that's being performed, the type of information and results that could come out of the test, and ultimately are really intimately involved in reading through the report and then sharing the report and information with not only clinicians and other healthcare team members, but also the patients themselves. There's multiple ways to classify detected mutations, and here we're highlighting examples of how things are defined within the U.S. region. But in general terms, in literature, you'll find some of these terms to be similar: pathogenic versus uncertain versus non-pathogenic. You'll also find deleterious or suspected deleterious and non-deleterious as examples of different types of classifications of mutations. The challenge, though, is understanding how the bar is set to define what type of mutation is to be considered as pathogenic, likely pathogenic, which would be clinically actionable, versus of the U.S. versus known to be non-pathogenic. What I just mentioned there is sort of the key component. As we identify more patients, we have broader data sets, including our own data sets, that better understand the types of mutations that can be found in patients, and then connecting that with clinical activity or clinical benefit from therapies. We get a lot more information about the types of genomic alterations that are actually meaningful for evaluating and informing clinical care. Something that was determined to be non-deleterious or non-pathogenic 5 years ago, based on the data we have today, may now be considered something that is deleterious or pathogenic. And so we need to strive to try to keep up to date with those types of classifications for those variants. And one of the key pieces is that there's a lot of independent databases. Those are usually commercial clinical laboratories. When they run their own data, they are continuously cataloging and keeping track of that information to help inform in an iterative manner their own internal testing. And there's, of course, web-based databases as well. But one of the other key pieces is more of a public database. And that makes it a much more centralized database where everyone can help inform each other, and you have a larger data pool informing your variant decision-making. There was a joint consensus among molecular professionals, and they proposed basically a 5-tier interpretation and reporting standard. Myriad, a laboratory group within the U.S., responsible for BRCA analysis, which is one of our companion diagnostics within the U.S., currently reports using the terminologies deleterious, suspected deleterious, VUS, favor polymorphism, and no mutation detected. The joint consensus actually recommends utilizing pathogenic, likely pathogenic, VUS, likely benign, and benign. And you can see basically the definition on the right-hand side and how it ties into clinical practice. But even still, again, reinforcing this, that complexity that even with a consensus statement out there within the U.S., one of the larger tests, commercially available tests within the U.S. still is not necessarily matching up with that consensus language. Again, creating challenges for folks to interpret and understand the test. In particular, when you think about all the questions that we get around testing results, the variant of uncertain significance is really challenging. You may also see it referred to as the variant of unknown significance, but really it causes a lot of challenges and problems in trying to understand and interpret that. And a lot of times data may align with a deletion or a mutation or a change and put it as a VUS. And again, as I mentioned, as we gain more data, as more databases are shared and understand the implications for clinical practice, then the VUS may actually become defined and more better associated with clinical data. And then in that case, it would actually be assigned as deleterious or suspected deleterious or non-deleterious, or in that case, benign. So you can see all of this hinges on having a lot of shared data, informed decisions, and basically an iterative process to continuously inform which types of mutations and changes are truly clinically meaningful and need to be tracked and understood for each patient. This slide here is really helpful just to kind of give you a snapshot picture of a number of tests, particularly used in the United States from Myriad, Foundation Medicine, as well as Color and Ambry. The key points, though, is this gives you this table about the different names of the tests, the types of genes that are actually tested, whether it's BRCA1 and BRCA2, or it's a panel where multiple HRR tests or genes, are represented, and also the type of sample that you can utilize. Notice that a number of tests you could actually choose to utilize tumor tissue, blood, and or the saliva to capture the buccal mucosa. For some tests, it's very specific that you need to use the tissue test or that you need to use a blood-based sample. For our purposes, it's really important to understand some of the panel tests that exist where you're going beyond just looking at the BRCA1 or the BRCA2 mutations. We have developed and honed in on a test, if you will, using the Foundation One platform out of Foundation Medicine within the U.S. for a number of our studies. And regarding that, we focused on 15 specific genes and the homologous recombination repair pathway. You see those captured on the left as our AstraZeneca HRR15 gene panel. It is, again, run on the Foundation One platform. From Myriad, in collaboration with them, we've utilized MyChoice HRD, which is a homologous recombination deficiency assay. You see there that it's not only looking at the mutations and the SNPs, but it's also looking at loss of heterozygosity, et cetera. So really it lends a score. And that test looks and feels different than other BRCA tests like BRCA analysis, which we've mentioned previously. And then lastly, Foundation Focus is the companion diagnostics looking at the BRCA mutations, but also the loss of heterozygosity, and ultimately gives you a specific scoring as well. So collectively, these are sort of a different approach to tests, if you will, than just more of that classic germline test for BRCA1 and BRCA2. And importantly, these tests look a little bit more holistically at a multitude of factors, looking for either the cause of HRD, looking at those gene mutations, or the effects of HRD when you look at the different types of scores in some cases. Within the U.S., the FDA has 3 companion diagnostic tests currently for BRCA testing in ovarian and breast cancer to determine eligibility for treatment. I'm not going to go into the details here. They're listed here for you. But as we continue to move forward, there's, of course, the chance that this would need to be updated. So please take note of the dates and refer to any updates through NCCN as well as through our label, et cetera, to understand current FDA approvals for our tests. So now we're going to dive deeper into other components impacting the clinical implementation of the test that we just walked through and discussed. Some of the key points there were to really think about your multidisciplinary team. And in your region, it might look and feel different based on the healthcare system that you operate with and the access that patients have to different providers in their healthcare team. We'll touch base on that a little bit and just really kind of walk through potential healthcare team members for you to consider how they would integrate within your health system. Next, we'll talk specifically a little bit more about genetic counseling, and it may or may not apply as much to you, but we can still walk through it because in general, the impact from genetic counseling does still impact the overall worldwide approach to testing and how things are considered, especially in clinical trials. And there's additional information from the genetic counseling point of view that can still be understood for any healthcare team member who's actually the interface with the patient and their caregivers or their carers to actually help them understand the test. And lastly, it's important to note there are a multitude of financial barriers to testing, and while we won't get into the details about specifically reimbursement, it's important to understand from a medical perspective that with insurers, it's important to be able to demonstrate a medical necessity for the test, that justification for the specific testing approach. Are we going to be utilizing multiple tests, or are we looking for multiple genes or just a single gene, et cetera, because they're making the comparison across other types of tests that are out there. In addition, we and other pharma groups as well as advocacy groups may have patient support or access programs to help aid in the accessibility to testing and cover particularly financial costs. And then lastly, of course, we want to make sure that we have informed pathway or other key payer decision makers to really make informed decisions on coverage or recommendations in their pathways regarding how tests should be utilized and implemented in the clinic. So when we sit back and think about genetic testing, it's easy to understand that, of course, the medical oncologist, or otherwise we may call them the treater, the person who's really responsible primarily for the patient's care, it's easy to see that they should be involved in the decision-making process and understand the testing options available. But it's important to think a little bit broader because it's the surgical team or the interventional radiologist in the healthcare team who may be able to have access to a specific tumor specimen. And so that's a really critical member of the team for actually assessing a sample if you're going to be doing tumor testing. The other component is the path department. Processing of the actual tissue itself is really important. It's very methodical and technical, so they need to be really well educated about the trade-offs for different types of preparations, how they handle the slides when they are selecting slides to send off for testing, how do they pick and identify those slides that have the best representation of tumor cells, as well as the best possibility for getting a successful DNA extraction is really critical. And in addition to that, the complementarity, if you will, if you have only one path department, is the molecular pathology component. sometimes you specifically have a genetics department, but really it's those individuals who understand the genomic testing component of what happens in your clinic and can really weigh in and not only at times actually conduct tests on site in a local lab, but in addition to that, weigh in on the decision-making of what types of tests are offered and available to treating physicians in the particular practice. Next, we see mentioned clinical genetics. So there are different specialists, and they may go by different titles in your particular healthcare system, but there are certainly specialists who really particularly focus on the genetics that impact our health. And so in particular, germline testing is really tied to their interest and their specialty. And germline testing in parallel or with consecutive to tumor testing, the counseling by these clinical genetic specialists is really critical for not only informed consent potentially, but also critical, of course, for understanding the results of the test and the implications across family members. And then as mentioned at the bottom there, we have the oncologist or the treating physician, and they need to really assess that relevance of the test to impact their treatment decision. But what you notice here is there are a lot of potential people involved, a lot of stakeholders, and they all own sort of a piece of not just a decision-making component, but really need to have a better awareness and understanding of the types of tests that are available in their specific market and their specific region, and the actual fit for purpose approach. What tests that are available will help address which clinical questions, and how can I best utilize the testing options in front of me to answer the clinical questions for each particular patient. Each tumor type, again, you may be weighing between being able to have access to tumor tissue or not. You may be weighing someone who's a very frail elderly patient. Being able to do even a blood draw for them might be challenging, so it might be easier than to go with a buccal mucosa swab. So all of this multidisciplinary team is really weighing a whole bunch of factors case by case, but they're also making decisions at the clinic level, and then sometimes, of course, these are our informed decision makers informing across at the regional level or globally worldwide. So it's important to consider testing in a very multifaceted way and looking at this broader multidisciplinary team. When you get into specifically genetic counseling, and again, the level at which this impacts your specific region, you'll have to think through carefully. But it's good to call out kind of some of the up-and-coming things that we see for genetic counseling. And one of those is that the counseling can be done in person, by telephone, or also video-assisted. So we are seeing, because of the challenges of having so few people and specialists who are focused on genetic counseling available, the patients are now getting access to genetic counseling services through either an app or a video that's run, for example, on an iPad in a clinic. And really, it's to kind of set up and queue up things for further discussion with the healthcare providers, but it can walk through a whole bunch of potential topics. So genetic counseling can certainly walk through hereditary risk based on the personal family history, explanation of the specific tests that will be run, the implications of the potential results, whether they could be positive, negative, or non-informative, what would that mean, the psychosocial risk and benefits of the results, the real impact, if you will, to the patient beyond just their care, but also the broader impact of how they interact within society, the risk of passing that mutation to a child, or whether or not the risk there could be for another family member like a sibling, and then, of course, raise awareness and understand potential treatment options. One of the key points, though, is that a delay in testing could certainly delay treatment or really more limit or impact informed treatment decision-making. So one of the key things that you see depicted in the slide at the top in the gray visual is a referral for a pre-test genetic counseling visit. So when you need to see a genetic counselor in advance of the test, in the U.S., you may wait 4 to 5 weeks for that initial appointment. Once you have that appointment, then you also have to wait potentially for another appointment after you get the results. Then you still have to wait to get walk through those results in an informed way with a genetic counselor. Ultimately, it could potentially take 8 to 10 weeks to get a patient's BRCA test results to the provider in a way that they could then utilize to make a treatment decision. And for some tumor types, that would actually be the duration of time it would take potentially for even a patient to have received therapy, and they may have progressed now at this point or have passed away. The second component you see is the idea of having an urgent pathway. When you know there's an urgency around a specific timeline, in the case of, for example, pancreatic cancer, where patients are often diagnosed with very advanced disease, over 80% of, or about 80% of patients are not even surgical candidates at that point because of the advanced level of disease that they have. They wait less time, but it's still 1.5 weeks to wait for that appointment. And then it still can take up to 3 weeks to get results. So ultimately, you're still looking at 3 to 4 weeks to get those patients the BRCA results to the provider, even with an urgent appointment. So of course, you can see that there are a lot of challenges here, and there's a lot of delay that may come in these getting the test results, especially when genetic counseling is required and or needed. And so that's one of the challenges that we face, and in particular for germline testing, genetic counseling is absolutely critical for the understanding of the ramifications for family members. Now you can see kind of in this table a breakdown and comparison between germline testing and tumor tissue testing. We already touched on the fact that we are in this paradigm shift for predictive testing and the reason for testing, which is really important, and understanding that germline testing is the way to understand that predisposition or familial risk. There are advantages, if you will, for tumor testing in that you would not need a genetic counselor depending on the rules and regulations in your area. But the other advantage for germline testing would be because it's buccal mucosal swab of the saliva or by blood-based, you have a non or minimally invasive sampling procedure. The results are highly accurate and reproducible, et cetera. The limitations, though, with germline testing, you're not going to identify patients with somatic mutations, and there could, of course, be a lot of delays due to the access to genetic counseling. And with tumor testing, the limitations are you cannot easily distinguish between germline and somatic mutations, so you won't get that from the results. And you may have to then, of course, refer a patient who has a positive result for a germline test. And also because of the sampling issues, there may be either spatially or temporally variable information due to the nature of somatic mutations in the tumor sample. And all of these factors weighed, heavily with the healthcare team, ultimately result in sort of a couple of options to think through. The healthcare team is going to make a decision. Do they want to do a standalone test? Are they going to decide to only do germline or only do tumor testing? Will the healthcare provider and or the clinic decide to do some sort of sequence to the testing? Will they start with germline and then reflex to tumor, and in what circumstances? And then the vice versa. Will they start with tumor and ultimately order a reflex to germline? And again, in what circumstances will they do that? Will that be only when there is a positive result, or will that just be based purely on time because of access to a genetic counselor? The other idea is, do the tests get ordered literally simultaneously? Do they really truly run in parallel, where you're taking the tumor sample and you're taking the blood or saliva sample and submitting those, if you will, in tandem? You're triggering both testing processes at the same time, understanding that based on the technology, based on the access to genetic counseling, the tumor results will likely come in, or will, come in sooner, but knowing that you have triggered and you will understand both the tumor test, which captures germline somatic mutations, but you will ultimately at some point have also the germline test to really confirm for you what the germline status is. And in closing, you can see that the world of testing can be very, very complex And we stay certainly grounded in our data and understanding of what we did within our clinical trials. We know our protocols, we know our laboratory manuals from what we did in our studies, we know our indication statements, and we also understand patient selection and criteria and whether or not we had a true companion diagnostic that's filed from a regulatory perspective or within our region if we're going to be looking at LDTs, for example. So we know some very concrete, specific information that helps us in our quest to help educate and inform the healthcare teams and other key decision makers around the key information they need to make decisions about testing. But you can almost juxtapose or have a challenge here once you get into the real world. When that data comes outside of the clinical trial setting, there are a lot of challenges to implementing testing in the real world. It's important that clinicians and healthcare team members really understand the purpose of the test. You know, are they trying to do a test to assess familial risk, or are they trying to do a test to inform clinical treatment decisions? Or are they, you know, when they look at that information, are they really not even thinking from a treatment decision perspective, but more of a research question, not just clinical trials, but other information like prevalence of mutations, for example. So the purpose of tests will really matter and come into play and filter how the healthcare team processes information about testing. The next piece, when you think beyond the healthcare team, in particular around the med-onc, but you really think about the multidisciplinary team, the broader multidisciplinary team that we discussed earlier and kind of went very stepwise into all the potential members, you really need to understand the genetic counseling component, what that looks like within your region, and it may not be done by a specific certified genetic specialist, but that same function, if you will, may be carried out by other members of your healthcare team. And at the end of the day, the real key piece here is that you are having a multitude of stakeholders that really play a role in making sure that testing is done and completed for patients, but also that patients can better understand their results and the implications for their family members. The other piece we touched on a lot earlier is just the concept of having an approved test through a regulatory pathway, something that's tied specifically to our drugs or any particular drugs, like an FDA-approved test. It's listed in the indication. But there are also other tests as well. The LDTs that may capture and test for similar or same information, but they may not be the exact same test. There's also the challenge of navigating commercially available tests versus tests only available in the research setting that may still provide information, but how will a healthcare team utilize that information? The test characteristics matter. The sensitivity, specificity, the analytics around the test matter. That turnaround time of the actual test itself, once, you know, the sequencer is started, how long does it take to get the results from the machine matters, and of course the sample requirements. And that also goes into pre-analytics for how the samples are prepped as well. The guidelines, pathways, and payer coverage certainly impacts access to the test and also the order of testing that could be completed and also the application of results, and really can be a very key driver to how widely available testing becomes in any given region. If there's not a lot of support at the guideline level or other, you know, gold standard setting criteria that really say that the standard of care should be including testing, then it may not be included or implemented into real-world clinical practice. And of course, one of the key components here is really making sure that patients do have an informed consent and any of their concerns are addressed so that they understand the implications of the testing that they're going to have done, and not only implications for them, but their families as well. And indeed, for some patients, we know that often they can refuse to have genomic testing because of the concerns of having genomic information about themselves and or their family members. And lastly, some of the real-world concerns, of course, especially when we think about, if you will, quote, big data, as it can be referred to, when you have a large data set and we get a lot more learning data sets, and our data handling capabilities become even more and more complex. One of the key questions, of course, also that comes up is the idea of who owns the genomic data. So you can see here that while we anchor ourselves in understanding our clinical trial data and our indications and our labels and our regulatory status within our regions and worldwide, when we communicate about our interest in HRR testing, we run into a lot of challenges of how testing will be implemented within the real world, especially across different stakeholders, and weighing the interest of the patients, and really ultimately what is going to be helpful to inform clinical practice. So in conclusion from today's discussion, I just want to make sure that I highlight to feel welcome to go back through and look at a lot of these slides have a lot of detailed information that we did not walk through reading out each word today. We also have a lot of references included, and those will be really helpful. And in addition, there are a couple of backup slides included after this one that gives you some additional key points around ctDNA to consider, and then an additional section of backup slides around genetic counseling, and walks you through the ENGAGE study that we've helped conduct to really better understand the implications of genetic counseling and utilizing different methods to help genetic counseling reach the patient. And then final conclusion, I want to thank you for your time, encourage you to reach out to your local biomarker team expert or other colleagues. If you have any questions, we welcome those and are welcome to talk and sit down with you to discuss this in more detail. And also, if you should have any other additional questions as you're out there utilizing the information, we welcome any feedback as well. Thank you again for the time. We hope this was really helpful for you today. And again, just encourage you to go back and feel welcome to utilize the additional resources, references, and your local biomarker expert for any questions you may have. Thank you.